Hey guys, so we're going to be talking about transcription and RNA processing today. So when we look at our central dogma, which is DNA to RNA to proteins through transcription and translation, we're going to be looking at this first piece here, DNA to RNA through transcription, and then the mRNA is going to get processed or modified before it leaves the nucleus. So gene expression is the production of a protein from the instructions on the DNA. Proteins have many functions in our body, and it requires several steps to get from the DNA to that protein. Again, it's going to be the central dogma. DNA to RNA through transcription, RNA processing, and then from our RNA to protein through translation. Now, transcription or transcribing. Transcribing is going to be recopying. Scribe means to write. So we're going to be rewriting the instructions into mRNA to be sent out for translation. Again, in DNA to RNA through transcription, that's going to be found in the nucleus. RNA processing also happens in the nucleus because we have to prepare that mRNA and protect it so that when it leaves the nucleus, it's protected and that it can carry the information to be translated. Messenger RNA is the instructions on how to build the protein. Now again, it is not the DNA. It's a copy. It's a copy that can be used and then degraded while the DNA, the original, the clean set of instructions, is held safe in the nucleus. So the messenger RNA is sort of like blueprints that you actually take to the work site. They might get some coffee spilled on them. They might get some rips, tears here and there. You might have to write on them. But you have the blueprints back in the office that are pristine and safe, and that's going to be the DNA. So they're the instructions on how to build a protein. They're produced in the nucleus. They carry information from the DNA to the ribosomes, and they contain codons, which specify particular amino acids to build the protein. Now, what is a codon? It is a set of 3 bases in the RNA that encode for a specific amino acid. So here's a codon, AUG, here's another codon, UUC, another codon, and so forth. UAG is an actual very special codon. It is the start codon. It starts every single gene or mRNA. And this codon actually encodes for a start methionine. Methionine is a type of amino acid. And methionine will start every single protein. Now, a reading frame is how we actually read these. We read them in sets of 3. And this will be important in translation because the tRNA reads this reading frame in the sets of 3, or the sets of codons. Now, the mRNA is created off of the template strand of the DNA. Now, remember, DNA has 2 strands. The template strand will actually be used to encode the mRNA. Now, the mRNA will be complementary to this template strand. But remember, mRNA does not have thymine. mRNA has uracil. So wherever you want to put a thymine, you have to put a uracil. The other strand of DNA is known as the coding strand, and it will look exactly like the mRNA strand, except for the T's and the U's. So again, the coding strand will look like the coded mRNA. Now, there are some key features or key players for transcription. We've got the promoter, which is a segment of DNA above the gene that we want. And it basically says, this is the start of the gene. This is the start right here. Transcription factors find and bind to the promoters for specific genes to be copied. So they choose which genes are going to be copied. Then they help align the RNA polymerase to the start of the gene. Now, what is RNA polymerase? It lays down new complementary RNA to the DNA template strand. Polymerases, in general, are going to lay down bases. Now, what comes in front is going to tell you what type of base. RNA polymerase actually lays down RNA bases. A DNA polymerase, though, would lay down DNA bases. So, RNA polymerase is going to lay down RNA since we're making mRNA. Enhancers can sit on the promoter and make it easier to find. It's like a big billboard or a sign that says, right here, come to this gene. So how does this work? We start off with initiation. Transcription factors bind to the promoter region, help grab and align that RNA polymerase to the start of the gene, and the RNA polymerase unwinds the double-stranded DNA and creates a transcription bubble. Please remember that an enhancer can be sitting on that promoter. Next, for elongation, now that we have this transcription bubble, the RNA polymerase runs along the template strand, laying down complementary RNA, making the mRNA, or the messenger RNA, right here. Termination. Eventually, the RNA polymerase will find a site, or a termination site, that indicates the end of the gene. The RNA polymerase will stop and be bumped off at that site, releasing the RNA. Here's the full view of initiation, binding to the promoter, elongation, making the transcription bubble and actually adding complementary RNA, and then termination. It's bumped off at the site and releases the RNA. Now, what happens to that RNA? Right now we're in the nucleus. Before it can leave, it must have some modifications done. We add a 5' cap to the sequence, which is important for regulating translation. Who's going to read it? We add a 3' poly A tail, lots of A's to that end of the tail. Why? Because out in the cytoplasm we have nucleases. These enzymes break down any type of nucleic acid, which includes RNA. Now, if it takes a bite out of these A's, we're not going to have our actual message disturbed. We're going to keep it intact. And so it can chew on these A's for a while before it actually destroys our message. And so we want to protect our message. Finally, we have splicing. It's the removal of the junk DNA, or junk sequences known as introns. Introns interrupt our message. We don't want the introns, and so they're removed or spliced out, leaving the exons actually in our message. So what do we do to our strand of mRNA? We add a 5' cap, we add a 3' poly A tail, and we splice out the introns. And we have our message. And it can actually go out to the cytoplasm and be protected. Well, we've hit a site, which means we're terminating for today. See you later.